Welcome dear students, this is my cellular energetics lecture series. In my previous videos I have discussed about glycolysis, and we know that the end product of glycolysis is pyruvic acid or pyruvate. Now the pyruvate is transformed into acetyl-CoA and take part in the TCA cycle or tricarboxylic acid cycle. It is also known as Krebs cycle or citric acid cycle. Now in this class I will talk about how pyruvic acid or pyruvate is converted into acetyl-CoA. So basically the reaction that occurs is pyruvate, the end product of glycolysis. Pyruvate is actually react with coenzyme A and nicotinamide adenine dinucleotide cofactor, and this is actually irreversible reaction that produce carbon dioxide, acetyl-CoA and NADH plus H plus. So this is the intermediate step between this glycolysis and TCA cycle. The pyruvic acid or pyruvate is actually transformed, changed into acetyl-CoA. So how this reaction occurs, we will learn about this in this class. So I have mentioned that in the glycolysis 2 pyruvic acids are produced. So there should be 2 coenzyme A, 2 NADA plus. So 2 carbon dioxide will be released, 2 acetyl-CoA will be generated, 2 NADH plus and 2 H plus. So this pyruvic acid actually 3 carbon molecules. Now the acetyl-CoA contain, this acetyl group contain 2 carbon, and for this reason 1 carbon dioxide actually released. The reaction that is taking place here is called oxidative, remember this, oxidative decarboxylation. Why it is called oxidative decarboxylation? As we can see here, CO2 is released, so this is called decarboxylation. The reaction where carbon dioxide is released is called decarboxylation, and oxidation also takes place here. I will show you in details. Now the enzyme that perform this function is called pyruvate dehydrogenase complex. So the enzyme name is pyruvate dehydrogenase complex. This complex actually multi-subunit enzyme proteins, and it contains different catalytic enzymes and regulatory enzymes. I will tell you, but at first you have to understand this reaction is actually take place in the mitochondrial matrix. So as I have mentioned that glycolysis occurs in the cytoplasm. So the pyruvic acid or pyruvate should be present in the cytoplasm and it should reach to the mitochondrial matrix. How it pyruvate enter inside the mitochondria? There is, let's assume, suppose this is the mitochondrial membrane. There is a symporter. There is a symporter called H plus pyruvate that actually pump hydrogen ions and pyruvic acid inside, and it actually take energy. Now from the cytoplasm, pyruvic acid or pyruvate enter inside the matrix, and this enzyme complex is present in the mitochondrial matrix, and it actually take place the cause these reactions. Now the basic fact about this complex is that this complex have 3 catalytic enzymes and 2 regulatory enzymes. This pyruvate dehydrogenase complex is actually multi-subunit proteins, but these 3 catalytic enzymes and the 2 regulatory enzymes play essential role in this reaction, but there are so many subunits are present that actually support the structure of this complex. Now the name of these catalytic enzymes are called E1. The full form of E1 is actually pyruvate dehydrogenase. There is another catalytic enzyme called E2. This name is dihydrolipoyl trans-acetylase, and the third catalytic enzyme is called dihydrolipoyl dehydrogenase. And the regulatory enzymes are, the name of the regulatory enzymes are pyruvate dehydrogenase kinase. Another regulatory enzyme is called pyruvate dehydrogenase phosphatase. So there are actually 5 enzymes that making this complex functional. Now you have to remember another important fact, that is these 3 enzymes are associated with cofactors. These cofactors play an important role in these reactions. Pyruvate dehydrogenase associated with a cofactor called TPP. The full form of TPP is, I am writing here, TPP means thiamine pyrophosphate. Do you know this TPP is actually nothing but vitamin B1, that is thiamine. So it's actually derivative of vitamin B1. Now the dihydrolipoyl trans-acetylase, the cofactor is lipoic acid and coenzyme A. These 2 are the cofactor for this enzyme. And the FAD and NAD plus are the cofactor of this enzyme. Now this FAD is actually a derivative of vitamin B2, that is called riboflavin. This NAD plus is actually derivative of vitamin B3, this is called nicotinic acid. And this coenzyme is also a vitamin B called vitamin B5, this is called pantothenic acid. So these are all actually derivatives of vitamin B. Now that react, okay, now I actually tell the function of this regulatory enzyme, then I will go to this catalytic enzyme and the main function, how this actually reactions occur. The kinase enzyme actually inhibit a particular enzyme of this complex called E1, because it phosphorylates the E1 at serine amino acid. You don't have to remember this. There are 3 serine residues that actually gets phosphorylated by this kinase and gets inhibited. And the opposite role is performed by phosphatase. So you remember that E1 is actually if the phosphate group is removed, so the E1 gets activated. So this is the regulatory function of these 2 proteins. Okay, now the 5 cofactors, 1, 2, 3, 4, 5. These 5 cofactors can also be categorized. One is called catalytic cofactor, that is associated with the enzyme. The example of these catalytic cofactors are TPP, lipoic acid, and FAD, flavin adenine dinucleotide. But the other 2, that is called stoichiometric cofactors, are coenzyme A and NAD plus, that actually comes from the outside but acts as a cofactor, but is a stoichiometric and is a catalytic. It is associated with the enzyme. Now what happens, I will show you here in this reaction center. Let's suppose this is a pyruvic acid, that is the CH3 C double bond O C double H, or you can write here C double minus, pyruvic acid. This is a pyruvic acid. Pyruvic acid or pyruvate, simply pyruvate, the end product of the glycolysis. So there are 2 pyruvic acid. These pyruvic acids actually decarboxylated by this enzyme pyruvate dehydrogenase, and it will release 2 carbon dioxide. The first reaction is the decarboxylation reaction, so it releases 2 carbon dioxide. Now these are the carbon source of this carbon dioxide. So this group actually remains, and one cofactor is actually accept this remaining part. This is called TPP, thiamine pyrophosphate. Actually take the remaining of this part, and the formula will be CH3 COH and then the TPP. So TPP accept this. This is called hydroxy ethyl TPP. Can you see here? This group is actually transferred to the TPP. Now this TPP is actually the cofactor of E1. So this thing is occurring in the E1 enzyme. Now to actually convert this hydroxy ethyl TPP to TPP, that means thiamine pyrophosphate, another enzyme, or actually the cofactor actually take part. This cofactor is called lipoic acid. This is the functional part of lipoic acid is called lipoamide. This lipoamide is actually oxidant or oxidative conditions. Oxidative conditions. Remain oxidative conditions. Okay, lipoamide. This is the part of the E2 complex. So at first carbon dioxide is released. Now the making of acetyl-CoA, how it occurs, I will show you. So lipoamide actually take this part. So at first TPP take this part, then lipoamide take this part and make acetyl lipoamide. Acetyl lipoamide. I have mentioned that this E2 also have a cofactor called coenzyme A. So coenzyme A actually comes and take this acetyl group and make acetyl-CoA. 2-acetyl-CoA. So all these things are 2. Okay, you have to understand this. There are 2 pyruvate. There should be 2 acetyl lipoamide. Okay, so there should be 2 acetyl-CoAs. There should be 2 coenzyme A. Now when coenzyme A takes this acetyl group, then the lipoamide is actually relieved. So lipoamide, but this lipoamide is in reduced in conditions. Reduced in conditions. Why it is reduced? Because lipoamide has some thiol group, SH group, 2 SH group are there, and it remains as reduced conditions. Now another enzyme complex come into play. This is called E3 complex. In the E3 there is a FAD. Now the FAD actually take the hydrogen and helps in the transformation of this reduced lipoamide to oxidative lipoamide. So the arrow should be here, from here. So FAD actually transform into FADH2. FADH2. You have to understand this. Okay. So this is the 3 catalytic enzymes, regulatory enzymes. You know that this kinase inhibits, phosphatase activates. How this kinase inhibits? Because this kinase gets activated by ATP, okay, coenzyme A, and this phosphatase is actually activated by insulin. So by this way, this enzyme, actually the phosphatase activate this enzyme, and the reaction takes place by a separate way. Like this pyruvate actually at first decarboxylate, means release carbon dioxide, then this TPP actually remove this part to the lipoamide and make it acetyl, then coenzyme associate with the acetyl-CoA, and now the FAD, that is the part of this hydroxylipoyl dehydrogenase. I have mentioned here that the catalytic cofactor are the TPP, lipoic acid and FAD, and the stoichiometric cofactors are CoA and NAD. Now the FAD becomes FADH2, but it should be converted into FAD. It should be converted into this, because then if the FAD becomes FADH2 permanently, then this reaction will stop. So this should be recycled back, and now the last cofactor NAD plus take part. So NAD plus actually take the hydrogen and make NADH, the H plus. So this is the whole reaction. Pyruvate coenzyme NAD, 2-acetyl coenzyme, carbon dioxide, and so here this reduced lipoamide is actually gets oxidated, so this is called oxidative. And also the FAD is actually from the reduced to oxygen formed by the NAD plus, so this is actually oxidative decarboxylations. So the main moral of this reaction I will tell you here. The main moral, the second part of this cellular energetics is the conversion of pyruvic or pyruvate to pyruvate into 2-acetyl-CoA, and it actually we get 2 NADH electron carrier that actually helps in the electron transport chain reaction and generate proton motive force that indirectly helps in the ATP generation. So it is actually, it will help this NADH. So we are taking this count. So this is the complex. Okay, I can give you another information that arsenic can poison this lipoamide or lipoic acid at the thiol group, that means SH group. This arsenic can bind with the thiol group and inhibit its cofactor as a cofactor. So by this way arsenic can poison. Okay.